So I hope you can all hear me and you can hear my voice. Please let me know anytime that you can't hear my voice, right? And thank you for joining today because we were having problem yesterday in connecting. I was facing problem in connecting Zoom. Um, I don't know what happened, but it was not working. Okay, so we were discussing about biomass yield and how this biomass yield, basically it is helping us understand a correlation between different substrates and different elements that are being used, right? So, and particularly that they are being used in the media, which is being used to set up the fermentation. So, this biomass yield, it gives you an idea about a positive or a negative correlation, whether the substrate added, reduced, or whatever is going to increase the biomass yield, or is going to increase the overall biomass, and it's going to increase the product of the fermentation or not. So that yield coefficient is very important in order to draw an estimate and in order to figure out how the substrate is affecting, or how the substrate is behaving inside once it is put into meat. So there are different other elements which are playing their role, like different operating conditions, varying pH and temperature. They can alter the figure of your biomass yield. And not just because what you were considering in biomass yield was the substrate, but then there are other factors that we know that equally affect the microbial growth, that is temperature, that is varying pH. All of these has an important role. And moreover, what kind of species that you're using, different strain can behave in different environments, and different individual can behave in a different environment, like what we have in the second point over here of Saccharomyces. Um, and what this Saccharomyces is doing, that this is a biomass yield coefficient, and it, it is actually varying from aerobic to anaerobic conditions, right? So it is slightly higher when there are aerobic conditions, and quite lower when there are anaerobic conditions. So what it shows is that aerobic conditions are kind of favoring the overall biomass yield, right? So, um, okay, so what we're talking about that, okay, a little bit in Urdu for you people. So, अब इसका मतलब क्या है? इसका मतलब ये है कि जब आप फर्मेंटेशन मीडिया सेटअप करते हैं तो सिर्फ सबस्ट्रेट, जैसे कि आपने कोई एलिमेंट डाला, कार्बन डाला, आपने कोई और न्यूट्रिएंट डाला जिसमें नाइट्रोजन सोर्स थी, इसके अलावा भी देर आर फैक्टर्स दैट आर प्लेइंग द रोल, राइट? जैसे कि टेम्परेचर, आप सबको पता है कि जब एक माइक्रो ऑर्गेनिज्म ग्रो करता है तो पीएच भी मैटर करता है, टेम्परेचर भी मैटर करता है, और उसका जो एनवायरनमेंट है वो भी मैटर करता है। तो सिर्फ ये नहीं है कि आपने मीडिया में कार्बन डाला या नाइट्रोजन डाला या उसको कौन सा वाइटामिन दे दिया, इसके अलावा भी फैक्टर्स हैं, और वो फैक्टर्स जो हैं जारी बात है बायोमास यील्ड को इफेक्ट करेंगे। And different from this is an example in the second point, and what this example in the second point says कि एक Saccharomyces cerevisiae is the species of the fungi which is the species which is being used, और उसमें दो किस्म के कंडीशंस दिए, एक एरोबिक एंड एनारोबिक कंडीशंस दिए, एंड दे वर ट्राइंग टू एस्टिमेट कि कौन ज़्यादा फेवरेबल है। अब उन्होंने बायोमास यील्ड कैलकुलेट किया, और बायोमास यील्ड कैलकुलेट करके ये देखा कि 0.56 जो है, विच इज़ स्लाइटली हायर फिगर, वो आता है एरोबिक कंडीशन में, और फिर 0.12 आता है इन एनएरोबिक कंडीशन। तो जाहिर बात है कि यहाँ जो सर्कमस्टांसेस और इन्वायरमेंट है उसमें एरोबिक इज फेवरिंग द बायोमास yield एंड द बायोमास, राइट। तो सबस्ट्रेट्स के अलावा जो आप मीडिया में डालते हैं लाइक कार्बन एंड एक्सेट्रा, उसके अलावा आपके इन्वायरमेंटल फैक्टर्स, पीएच, टेम्परेचर, उसका ऑक्सीजन अवेलेबल है, ऑक्सीजन लेस अवेलेबल है, ये सारी चीज़ें ओवरऑल बायोमास में अफेक्ट करेंगी, और बायोमास ओवरऑल एक फर्मेंटेशन को अफेक्ट करेगा। Okay, so what we're looking into in next 3 slides, 2 to 3 slides, is different kind of problems that are encountered once you're setting up a fermentation, and they are actually based on common sense, right? They're not much they're not very complicated or very complex to understand. They're very simple consequences that has to be kept in mind, right? So like we're talking about that elemental requirements for microorganism is important, and so there is a lot of emphasis which is made on improving the nutrients and nutrient source and the media to improve the overall rate of fermentation. But then there are other kind of problems that can adversely affect fermentation, and that can adversely affect the biomass you're trying to achieve. उसमें point number 3 is, for example, those that are rapidly metabolized may repress product formation, right? अब इसमें क्या बताया गया है, इसमें ये बताया गया है कि there are certain kinds of nutrients और certain kind of compounds जो कि बहुत जल्दी यूटिलाइज हो जाते हैं, मतलब माइक्रो ऑर्गेनिज्म फेवर करते हैं और वो बहुत जल्दी मेटाबॉलिक पाथवे से यूटिलाइज हो जाते हैं। Those nutrients which are very actively utilized, वो आखिर में प्रोडक्ट बनने में it can cause a hindrance, meaning वो प्रोडक्ट को सही बनने नहीं देंगे क्योंकि न्यूट्रिएंट बहुत जल्दी मेटाबोलाइज हो गए, राइट। तो आपने बायोमास तो देख लिया कि किसी न्यूट्रिएंट से अच्छा आता है, लेकिन आपने ये फैक्टर नहीं देखा होगा कि वो कितनी जल्दी मेटाबोलाइज होके कितनी जल्दी खत्म हो जाता है और उससे फर्मेंटेशन आखिर रुक जाएगी। ठीक है जी, सो दिस इज ऑल्सो एन इम्पोर्टेंट फैक्टर। Then the last point on this slide is to overcome this, intermittent or continuous addition of fresh medium may be carried out to maintain a relatively low concentration that is not repressive, right? Acha, what it's trying to say, it's trying to give you a solution of this point over here. What it's trying to say, it's trying to say that add fresh media after some time, right? थोड़ी देर बाद अगर आप फ्रेश मीडियम डाल देंगे तो वो जो बहुत रैपिडली मेटाबोलाइज होने वाले कंपाउंड्स हैं, it will stay around, and once it will stay around and it will stay around in low concentration, it will not repress the fermentation process. Then moreover, certain media nutrients or environmental conditions may affect not only the physiology, chemistry, but also the morphology of microorganisms, right? Now, what it's trying to say that the kind of media you put doesn't just naturally affect the biochemistry or the metabolic pathways. It is also playing a very important role in the physiology or morphology of the microorganism. What is morphology? The structure of the microorganism, right? This point number 3 over here, it tells you what kind of change can it bring, right? So it's saying that there's certain form of yeasts which are single cell, but in certain kind of media, what happens that they develop mycelium. Mycelium is those thread-like growth of fungi that usually shows, but yeast cells, they do not show mycelium growth, right? So these are pseudomycelium, and they are there because certain kind of media can cause morphological change. तो वो जो yeast जो single cell है, वो एक किसी किस्म के मीडिया में जाके माइसीलिया की ग्रोथ शो करता है, वो थ्रेड लाइक स्ट्रक्चर ग्रोथ करता है, विच इज एक्चुअली अ फॉल्स स्ट्रक्चर, because yeast is a single cell organism, right? तो फिर होता क्या है, they flocculate, they form clumps, which a yeast cell, which is a single cell, वो evenly distribute होता है एक liquid media में. लेकिन अगर उसके mycelia बनेंगे तो वो flocculate करेंगे, वो clumps form करेंगे. Similarly, they're talking about an exactly opposite example, and they're talking about a filamentous fungi may form pellets, right? So, वो जो आपका single cell है, actually these are opposite features talking about, right? Your yeast, which is a single cell organism, it actually forms pellets, and a mycelia growth comes. They actually form flocculates or filamentous fungi, right? So what's happening that single cell yeast is developing mycelium, and your filamentous fungi, they are behaving as single cell organism, right? So what is this? This is a morphological change in microorganism because of the media being used, right? So the third point, it says that such kind of changes might not affect the physiology, and such kind of changes might not affect the biochemistry of the cell. And you have to suit your situation. You have to see what kind of fermentation you have set up, and whether the change of morphology may be desirable and it may not be desirable under certain circumstances. Then continuing the same kind, the media adopted also depends on the scale of fermentation. So for small scale laboratory fermentation, few chemicals may be used, right? So this is something we've been talking about in the introductory lectures as well. And we were talking about that usually there are small scale fermentations being set up, and if there are small scale fermentation, we are not we are using quite pure chemicals because the overall volume is less, and it's easy for investors to buy expensive and well defined and refined chemicals to bring about their product formation. But if you are operating things on industrial scale, and if you are using large scale laboratory fermentation, then looking into cost of the media becomes something very important, right? So simply due to cost and the expenditures that are there in order to set up a very large scale fermentation, it is not possible to use very pure chemicals. So what do they use? They use actually different kind of alternatives, and in this lecture we are going to cover those alternatives that are there, right? So what are their cost effective nutrient sources? What are these nutrient sources, right? So usually these are carbon and nitrogen sources, and they are undefinable basically. And what do they mean by they're undefinable? Because they are used as raw materials. They are not like pure chemical. Like इस तरह नहीं होता कि आप सोडियम क्लोराइड का सिर्फ प्योर सॉल्ट यूज कर रहे हैं, या किसी नाइट्रोजन कंपाउंड में एक any one particular kind of amino acid you're using that. When the industrial scale fermentations are being developed, and the media for its use is being developed, so the carbon and the nitrogen sources um, उस वो डिफाइंड नहीं होते कि ये अमाइनो एसिड है, इसमें ये प्रोटीन है. They're just raw materials, and how we're going to see later on, right? So most are derived from actually either they are animal products or they are plant products. Often um, they come as a waste effluent from different other industries, or they may be as byproduct of other industries, and they usually vary in composition as well, because there is no standards, there is no defined particular nutrient source, right? So what happens that the nitrogen sources and the carbon sources, They are undefinable, so the effects of such batch to batch variation are determined. So what happens that since you're not using a pure and since you're not using a pure amino acid, so batch to batch there will be variations, right? And that batch to batch variation has to be determined, and it is their responsibility, those who have set up the industries and those who are developing the product. But it does vary time to time, right? Okay, so how do they deal with this? How do they know that there are batch to batch variation, and what do they have to do with it? So their small scale trials are usually performed at each before a new batch for fermentation is set up. And once you perform that small scale trial, you will examine the product. And once you examine the product and you get the same kind of yield, then you can take that that raw source coming from different industry into your large scale industrial fermentation process. So let's look into different sources. There is an extensive list of different sources that are put into a media. Like we all know, carbon source is a vital nutrient for all forms of life, for every kind of cell. So whenever you want to provide a microorganism with the nutrients, there must and should be a carbon source. So there should be a carbon source, right? So in most fermentation, it also serves as an energy source, right? So carbon requirement may be determined from the biomass yield coefficient, of course, because what it was, we're going to repeat this again, right? So what is this biomass yield coefficient? Biomass yield coefficient gives you a positive or a negative correlation between a correlation of the substrates that you can put in the media, right? So if it's a positive correlation with a substrate, it means adding more of that substrate is going to give you a higher yield. And if you have a negative correlation with a substrate, that means adding this substrate is going to reduce the yield and it's going to reduce the biomass. Right, so whenever you have to determine the carbon requirement, you use the same measurement of biomass yield. So actually what it's trying to say, it's not just the carbon that you're using to determine the biomass yield. You're viewing the biomass yield for almost all of the nutrients, right? So each may be determined by conducting a series of batch culture experiments where the specific substrate is the only growth limiting media component and other nutrients are in excess. Okay, so there are different ways to determine how important a substrate is, and in order to determine how important a substrate is, what you can do that you can limit the specific substrate in the media, and all of the other nutrients are in excess, and you are looking into the performance of this limiting substrate or nutrient, and if it's affecting the fermentation and if it's affecting or if it's reducing and lowering the entire yield, then obviously this limiting substrate is something very important and it needs to be provided in excess. So there are multiple ways you can do and you can see. So let's now one by one, we're going to look into the carbon source and the raw carbon source, right? Ali Akbar is raising his hands. अच्छा जी आपका I understood. I understood you want Urdu in between. Okay. So, what are the carbon sources? Carbon sources is the byproducts you get from different industries, right? So you must have heard of molasses. So molasses basically है क्या? A byproduct of grain and beet sugar production. जहाँ आपकी शुगर प्रोडक्शन हो रही है आउट ऑफ शुगर केन, वहाँ एक दिस ब्लैकिश लिक्विड एंड ब्राउनिश लिक्विड जो कि एक बाय प्रोडक्ट में निकलता है, जो कि शुगर की फॉर्मेशन में यूज नहीं होता। तो शुगर इंडस्ट्री के लिए तो वो बेकार चीज़ है, लेकिन वो जो बाय प्रोडक्ट दैट सिरप जो एक बाय प्रोडक्ट की फॉर्म में है जिसको मोलासेस कहते हैं, that is a rich carbon source, right? And it's really a cheap carbon source. तो ये डिफरेंट बाकी जो इंडस्ट्रीज हैं जिन्होंने फर्मेंटेशन सेटअप करना है, वो शुगर केन इंडस्ट्री में से मोलासेस उठा लेते हैं, बल्कि बहुत सस्ते लाइक चीपर फॉर्म बड़े सस्ते दामों उठाते हैं। वाई? बिकॉज इट वुड बी कॉस्ट इफेक्टिव for the industry to be set up, right? So but what happens that it is a very rich source, ठीक है जी, and this material is the residue remaining after most of the sucrose has been actually crystallized, right? तो हमने ये ऑलरेडी डिस्कस किया है कि एक डार्क ब्राउनिश किस्म का एक सिरप सा निकलता है विच इज़ वेरी स्वीट एज वेल, इट इज़ रिच विद कार्बोहाइड्रेट्स। तो ये कार्बोहाइड्रेट्स हैं, तो ये कार्बन का सोर्स होते हैं इन डिफरेंट फर्मेंटेशन प्रोसेसेस। तो लेकिन लेट्स ब्रेक डाउन एंड लेट्स लुक इनटू द मोलासेस की कंसिस्टेंसी कितनी है। So carbohydrates is round about 50 to 60%, right? और उन कार्बोहाइड्रेट में सबसे ज्यादा कौन सा है? It's basically sucrose. You know, sucrose is the sugar. Then 2% within this molasses is the nitrogenous substances, right? So there is 50-60% of the molasses syrup that is carbohydrate, and then there 2% would be of the nitrogenous sources, ठीक है जी। और इसके अलावा क्या है? इसके अलावा आपके पास vitamins हैं, इसके अलावा आपके पास minerals हैं, ठीक है। So molasses, if you want to explain what molasses is a byproduct of a sugarcane industry, जिसमें 50-60% carbohydrates होते हैं. That's why it is a good carbon source. ठीक हो गया? So overall composition, it varies depending upon the plant source and the location of the crop and the climatic condition under which it was grown. Okay, the last point is emphasizing कि आपकी क्रॉप कौन सी है, उसकी वैरायटी कौन सी है, वो किस क्लाइमेट में ग्रो कर रहा है, उससे ये परसेंटेज इफेक्ट होती है. Alright, meaning there's a kind of a variety of sugarcane that grows in Pakistan, and then there's a kind of a sugarcane that grows in Finland, and both of these varieties in the end would give you a molasses with a varying percentage of these compounds, of varying percentages of carbohydrates, of nitrogenous substances, of the minerals and the vitamins which are being used. Okay, other than molasses as a carbon, there is also a malt extract, right? Malt extract can also be used as a carbohydrate source. Okay, so let's first of all look into what is malt. So malt germinated cereal grains that have been mined in a process known as malting, right? So basically malt क्या है? Cereal grains हैं जो कि germinate कर रहे हैं. Germinate क्या कर रहे होते हैं? They start to grow the roots and they start to grow the leaves. Once they start to grow the roots and the leaves, उसकी germination शुरू हो जाती है, right? But right at the start of germination, you stop the process and dry. You dry the grains, and once you this causing the cereal grains to germinate, and then you this entire process is known as malting. All right. Please can you
मैं आया था ना, कौन सी बोरी लेके गया था जो आपने 3 दिए थे, 5 की थी और 2 के थे ना, इसमें तो भी नहीं है, दोनों में नहीं है.